feel it's worth at least mentioning a little bit about the development of the heart. So the fetal heart versus, the adult heart. Because it's also just kind of nice to, it's important to understand this because this is actually at the root of some congenital defects and things that happen. So it's important to understand some of the big differences between fetal and adult circulation. At the core, what's the big difference in the job of the heart if you're in a fetus versus in an adult?
That you have the umbilical cord.
So what is the umbilical cord doing?
Deliver nutrients.
No, but it also supplies.
Right,
And
so,
oxygen.
Blood too. Yeah, oxygen.
right, so the umbilical circulation, which the fetus has, umbilical circulation, umbilical. This is basically, we've got the little fetus here and sending blood out the umbilical arteries to the placenta, where it picks up oxygen, the umbilical veins bringing it back. What is happening at the placenta? What is the placenta?
It's like a capillary bed
And
with
what
connective
happens?
tissue.
But there's something more critical happening there. There are capillaries in there for sure.
But it has arteries from the fallopian tubes, I think.
Not fallopian tubes, but just from the mom. It's in
From
the
the
lining of the uterus, and you have the maternal circulation coming in.
It's getting oxygen and nutrients from the moms.
Right, so the maternal blood is coming in, and we can have an exchange of oxygen and nutrients and wastes between the fetal circulatory system and the maternal circulatory system. Right, the blood doesn't mix or anything, but you can exchange materials. So this is where this little fetus is getting oxygen, because the fetus isn't breathing. Obviously, you're not breathing if you're floating around in amniotic fluid inside the womb. You're getting oxygen. It's where they're getting nutrients. You're obviously not like, chowing down on your like kind of goldfish crackers and stuff when you're kind of floating around in the womb. As well as wastes get given away there. So if we think about, especially given the function of this, I'm gonna mute some people here. If we take into account that umbilical circulation is taking care of oxygen and nutrients and stuff, what does that mean about the function of the heart in the fetus?
It's irrelevant at this point because it's getting, the functions are happening elsewhere in the placenta by the parent.
I mean, if the heart wasn't pumping, we would never have blood going out to the placenta and coming back and distributing oxygen and nutrients to the whole body, the whole systemic circulation.
It's the distribution of oxygenated and deoxygenated blood that it's not the same as an adult.
Right. So what is the point? Like, what does the left side of the heart do?
Pump oxygenated blood to systemic circulation.
Exactly. So that we need, that we most definitely still need. But what about the right side?
Deoxygenated blood.
Which going
to the lung.
To the lungs.
To the lungs.
To pick up oxygen at the lungs, which is not necessarily
happening.
So you're going to send a minimal amount of blood towards the lungs to just keep the tissue alive, because it's still cells that need. But there's going to be a couple of major like bypasses. There's going to be what we call the foramen ovale. So the blood that would have gone out to the lungs, out the pulmonary trunk, instead just goes back into the left, not that way, left atrium and ultimately left ventricle out the aorta. There is another thing, the little ductus arteriosus, where blood coming up here cuts across from the pulmonary trunk to the aorta. So basically, most of the blood that would have gotten shoved, pushed out towards the lungs, instead gets, again, right ventricle normally would push to the lungs, but instead goes out, but then gets short-circuited to the left side of the heart. And if it still comes out, it gets short-circuited from the pulmonary trunk to the aorta. So most of the blood that's getting pushed by the right side of the heart is still going systemic. So that's a big difference.
Does this have anything to do with why the fetal heartbeat is so fast? Or is that something else?
That's something different. But
Okay.
this is responsible for some congenital heart problems. If normally this hole here seals up after you're born. The foramen ovale becomes the fossa ovalis. Normally this little connector bypass between the pulmonary trunk and the aorta closes off. It becomes the ligamentum arteriosum. If these don't close off properly, then you're going to have inefficient heartbeats because you're going to be mixing oxygenated and deoxygenated blood. So, so like in the tetralogy of Fallot and stuff, there's different things that happen where these things don't transition properly from the fetal version to the adult version. The one other thing that's different in fetal circulation is because you are not having to eat and process your nutrients. We talked about in normal circulation, where does the blood go from the digestive organs before it goes into general circulation?
Hepatic portal.
Yeah, exactly. That whole hepatic portal system. You go from the digestive organs to a second capillary bed in the liver for processing, and then back into circulation. And in fetal circulation, you also have another, the ductus venosus, which bypasses hepatic circulation. And again, you don't need to know all of the details, but you should understand the basics. One is we got an umbilical circulation, which is going to the placenta to exchange materials with the mom, not mixing blood, but exchanging materials across the placenta. We're bypassing pulmonary circuit by having these shunts like this ductus arteriosus, the foramen ovale. And again, we're also bypassing the hepatic circulation as well, since we don't need to process all these nutrients because we're getting them pre-processed from mom. So anyway, I just wanted to put that in there for completeness before we leave the heart behind. Okie dokie. Blood vessels. Obviously, these are the way we transport the blood. Like I mentioned at the beginning of our unit on cardiovascular system, you have about 100,000 kilometers, like 60,000 miles of blood vessels getting blood to within fraction of a millimeter of every cell in your body. That being said, there are a few different main types of blood vessels that we should talk about. But first, I will talk about the generic blood vessel. So this is kind of a generic blood vessel, and we will talk about the differences between different kinds of arteries and veins and stuff as we continue. But the basic parts of a blood vessel Blood vessels are always lined with simple squamous epithelium. It gets the name endothelium, is its fancy name. This is going to be important when we get to blood clotting in a little bit. Like, how do your little platelets know that there's been damage to the blood vessel and that it needs to actually repair stuff? It's because it's seeing like connective tissue, collagen and stuff, instead of just seeing this wallpaper of simple squamous epithelium, this endothelium lining everything. Endothelium is also what lines the chambers of your heart. In your heart, instead of calling it endothelium, it gets the special name endocardium, but it's the same stuff. Basically, all of these, the passages where the blood is flowing, whether it's through the chambers of the heart or whether it's through the blood vessels, it's all wallpapered with this endothelium or simple squamous epithelium. The middle part, we call, sometimes it's like tunica media. Oh, I should also mention the other, so endothelium, AKA, is also called tunica intima. The intimate tunic, because it's the one that's right up against, rubbing up against the blood cells as they move through. So sometimes when you're talking about blood vessels, they'll talk about just intima. So intima or endothelium is just referring to that simple squamous epithelium lining the inside of the blood vessel. So you should, if you see this word in reference to a blood vessel, you should know it's just talking about that simple squamous epithelium lining there. Okay, now let's go to the tunica media. This is the wall, the main wall here. This is basically going to be smooth muscle and kind of elastic tissue. So elastic tissue to give the vessel some stretchiness, because it's pulsing and all that. Smooth muscle to alter the diameter. So let me be explicit here. The smooth muscle, I want to make sure, I'm kind of running out of space here. So this changes diameter. Not for pumping. So yes, there is muscle in the walls of your blood vessels, but it's not about pumping the blood around or doing anything like that. It's the heart's job to generate the main pressure, or that we talk about the stretchy elasticity of these large arteries that propels blood. But the smooth muscle is about adjusting the diameter, which is then going to adjust resistance or, adjust the amount of flow in one place or another. So when you think about the muscle in the wall of the vessel, think about it in terms of diameter. Don't think about it in terms of like propulsion or anything. The smooth muscle is innervated primarily by the sympathetic nervous system. Innervation. I am so not here. I'm realizing I'm gonna get rid of the endothelium here so I just have more room. Okay, that's better. So this smooth muscle innervated by sympathetic autonomic nervous system. And sometimes they say vasomotor, vasomotor fibers, vessel muscle. So, we talked about one of the lack of antagonistic things between sympathetic and parasympathetic was the smooth muscle in the blood vessels is primarily sympathetic. There are some cases where autonomic parasympathetic can control blood vessel diameter, like, in sexual arousal, the parasympathetic nervous system is in charge of vasodilation that results in erection and things like that. But for the most part, when you think about the vessels changing diameter, you think about it being controlled by the sympathetic nervous system. And there are obviously, and we talked about, there's lots of other things that control that smooth muscle, right? We talked about the chemical signals, like, levels of oxygen or carbon dioxide and things like that. And there's other things that we'll talk about, like histamine, things that are going to cause inflammation. Nitric oxide is a classic little messenger for vasodilation. There's lots of things in addition to this that will also be affecting that smooth muscle and changing vessel diameter. So the outside is just, connective tissue to kind of strengthen it. We call it the adventitia. Or it can also be called the tunica externa. It doesn't matter for this class. I'll add one last thing in here because it's kind of cool. Plus it does come up in pathological situations sometimes. How do these cells, these little smooth muscle cells, endothelial cells, they're cells that need their own oxygen and nutrients. How are these cells getting their oxygen and nutrients that make up the blood vessel?
Do they do the special type of, I forget what it's called, actually. Someone else probably knows. I forget the name of it.
Diffusion from the blood.
So if it's a small enough vessel, a lot of your vessels are really dinky. If it's a small enough vessel, they actually can get by with just diffusion from the very blood that is within the vessel itself.
Is it the second transporter thing? Is that thing?
No, it could just be diffusion, like gas is just diffused.
All right.
Yeah, and then and they're so another thing though, if it gets bigger and bigger, then obviously diffusion is not going to work. So then you actually get blood vessels that serve the tissue of the blood vessel. And they get the kind of cool name, the vasa vasorum. It means the vessels of the vessels. So a large enough blood vessel actually needs its own smaller blood vessels to serve the tissue of the larger blood vessel. Sometimes these are what you see, like if you've ever gotten like kind of an infection from bad, like poison oak or something, and you get those scary red lines going up your arm, it's basically where you start getting the vessels of the vessels all inflaming and stuff and kind of outlining the route of the vessels going up your arm and stuff. So vasa vasorum. So this is generic blood vessel. Now we'll look at the three main types. Let's start with the arteries versus the veins. So by definition, when we say arteries, what do we mean by arteries?
Vessels
Leaving the heart.
So these are the ones that are leaving the heart.
with oxygen.
And what's that?
Leaving the heart with oxygen.
Not necessarily. If you're on the systemic circuit, you're leaving the heart with oxygen, like through the aorta. But if you're in the pulmonary circuit, the pulmonary arteries are going to the lungs deoxygenated. They're actually going to pick up oxygen. So arteries can be red or blue, right? Depends if you're on the pulmonary or systemic circuit. Definitely, yeah, don't think of oxygen being a defining quality here. You know, pulmonary veins returning, your veins return from the heart. The pulmonary veins are bright red because they've just picked up oxygen. You know, the veins, coming back from the body, though, dumping into the vena cava, those are obviously deoxygenated. So this functionally is going to result in also kind of the anatomical differences. If we think about, if you're leaving the heart, you are getting the full brunt of the pressure of the ventricles contracting. So you're going to have a lot of muscle in the walls if you're not going to explode, or if you want to be able to control your diameter under the intense pressure coming right out of the heart, these are going to have much thicker walls. You know, I'll say thicker tunica media. Veins are going to have a thinner tunica media. Right? If you are on the veins, we're going to talk about this in more detail. But the basic idea is you've got the arteries coming in. You know, by the time you get to the smallest arteries, the little arterioles, you're maybe down to 40 millimeters of mercury. You go through the capillary bed. Then you're in like the little venules. You're down to like 20 millimeters of mercury. You know, by the time you get to the vena cava, you know, at the heart, you're basically at 0 millimeters of mercury. There's almost no pressure left. So if you're thinking about, you know, so here we're going to start, like we said, what the map was about 93 millimeters of mercury when we're coming right out of the heart, right? Remember, that's what we calculated if you were like 120 over 80. So the arteries are working with these higher pressures. Veins are working with much lower pressures. So in order to adjust the diameter, which is important in both veins and arteries, you don't need that much smooth muscle to fight this kind of pressure, whereas you need a lot of smooth muscle to fight this kind of pressure. Because the pressure is so low, and if, like I said, by the time you get arriving at the heart, the pressure is pretty much 0. You're gonna, if you're just relying on the pressure to keep the flow going, you're gonna be disappointed. What are ways, if the pressure is low, how can we try to get the flow continuing?
The skeletal muscle contracts, right?
Yeah, so let's before we go there, we are gonna go there, but first before we get to that flow, we talked about being blood pressure, you know, over resistance. If the pressure is really low, how could we try to keep the flow okay?
Increase the resistance.
You know, decrease resistance. How can we decrease the resistance?
By
Get vasodilation.
Or-
You know, just in general, having much larger diameters. You know, veins tend to have larger diameters because that has less resistance, means it's more likely. There's also going to be way more anastomoses. Those multiple paths to the same destination. You know, we talked about the arterial anastomoses in your arm with the radial and ulnar artery going to the palmar arch, but that's pretty rare. There's a few examples of these in the arterial side. However, in the veins, there are always many, many paths to the same place. You know, if I'm in the hand and want to get up, back up to my shoulder, I can go up the cephalic artery or the the cephalic vein or the basilic vein or the brachial vein, and they all go to the same place. So there's a lot more just multiple paths to try to get to some place. And then the veins have valves, one-way valves. So that's going to be important. What that means is as the blood is kind of slowly limping back on its way back to the heart, that the blood can't go backwards. Again, normally if we were in class, if you just push the blood down your arm the wrong way towards your hand, you'll see these little bumps showing up where the blood is backing up against the valves. Whereas if you push the blood up towards your heart, you'll see all the little veins kind of flatten out. So these one-way valves. And then what Stephen was mentioning, this idea that surrounding the vessels, they're going through all of your arms and legs and this and that. There's muscles everywhere. So there's all this skeletal muscle. Not the smooth muscle. Smooth muscle is in the wall adjusting the diameter of this thing. But here we're just talking about the skeletal muscle, like your biceps and your triceps and your quadriceps and your hamstrings, all these muscles that move your body around that are external to the vessel. And as they are flexing and relaxing, they're massaging the vessels. So if I have some blood here, let's make it yellow so it's easier to see. And then this muscle is kind of squeezing a little. This is going to get splooshed up to here. Once the blood is splooshed to here, it can't go back. And then as I keep kind of flexing and relaxing my muscles, this little blood gets splooshed further along. So this massaging, sometimes people call it massaging, sometimes people call it milking. This massaging or this milking by the external skeletal muscles helps keep the blood moving along the veins and making it all the way back to the heart. And then another thing that's going on . So your heart and your lungs are there in your thoracic cavity. And kind of spoiler alert, we're going to, in fact, hopefully today even, we're going to, although maybe not, we're going to talk about pulmonary ventilation, the way that you get air in and out of your lungs. The diaphragm goes up and down. The pressure in your thoracic cavity is going up and down. And you have these major veins coming back. The vena cava is right in the middle here. The ventricles and the atria are in here. So they're getting pumped, pulled back and forth as, with every breath you take, there is this pumping of blood back into the heart. So they call this the respiratory pump. And that's happening because the heart and these large veins are in the thoracic cavity, which is undergoing these undulations of pressure just that are driving your pulmonary ventilation, driving your breathing, inflating, deflating your lungs. So if there's ever some question about like, how is it that blood still makes it back through to the heart through these low pressure veins? It's like you got this respiratory pump going, you've got valves making sure it doesn't backtrack. What else? You've got this milking by the skeletal, again, the external skeletal muscle helping kind of milk and massage the blood. The one thing it's not is the smooth muscle in the vessel. The smooth muscle in the vessel is about just changing diameter. If you're going to try to do increased venous return or something, you might have to change the volume of your veins or something. All right, so we've talked about veins enough. Arteries, I'll just say briefly, there's two main kinds of arteries. So these are the ones, elastic arteries from their name. These are these ones that are very elastic, and these are the ones that are coming right off of the heart that are keeping your blood pressure from dropping to 0 during diastole. So this is like the aorta, the pulmonary. So their job is basically to be really elastic, to be able to stretch out during systole, to kind of store that energy, and then compress and continue to propel the blood during diastole, so you have your diastolic pressure staying high enough. So that's one type of artery, and again, they're pretty rare. These are just the really big arteries coming right out of the heart. More common are what we call muscular arteries. Arteries. These are like most named arteries. Your brachial artery, your radial artery, your femoral artery, all those, the main point of their structure is to adjust the diameter, which we saw is a critical part of both maintaining the appropriate blood pressure as well as, appropriate distribution of blood. So that's what muscular arteries. And then if you have an artery that's really small, they call it an arteriole. Exactly when it becomes an arteriole depends which book you read. It's usually somewhere around 1/3 of a millimeter or something. If you have a vein that's really dinky, it's a venule. So these are just kind of dinky versions of arteries and veins. What else do I want to say while we're here? Let me talk a couple of pathologies that happen with blood vessels before we move on to the capillaries. That's not spelled. I think that is right. What is an aneurysm?
It's abnormal when the wall of the blood vessel gets thinner, and
Yeah, so if there's some kind of weakening, we have some artery and somewhere, and there's the normal blood pressure and normally the smooth muscle in the wall of the vessel is able to hold it and adjust the diameter appropriately. But if there is a weakening in that wall, you start getting this ballooning out. So that's what an aneurysm is. Minimally, it can be bad because it's putting pressure on surrounding structures, right? Because it's pushing out against things. When does this get really bad?
When it ruptures.
When it ruptures. A burst aneurysm can be, can be instantly fatal. Like if you have an aortic aneurysm, like where your aorta, the main artery leaving your heart, all of a sudden bursts, basically within half a minute, you've basically dumped your whole blood supply out into your body. I had a student who was like doing some intake with somebody who suffered a aortic aneurysm in front of them and just watched them like blank out and then die like in a few moments. Aneurysm, like in cerebral arteries, can also be fatal. If you have aneurysms and they find it, they can do different things. They put little reinforcements to try to keep it from bursting. In the brain, there's other ways they have this thing, googly, these little coils they put in that make the blood kind of, there's different approaches, clinical approaches. If you have an aneurysm and you see it before it bursts, they go in and they try to stabilize it. Other issues that happen, like atherosclerosis, or the hardening of the arteries. In this case, you end up having in that tunica media, you get this proliferation of smooth muscle and fat. So this is what we call a plaque. And it's basically lipids and excess smooth muscle. Obviously, this is an issue. This is a problem. If this is the lumen of my blood vessel here, this is going to make it hard for the blood to flow. So, we've got this constriction. If it gets really bad, it can totally block the blood flow. Minimally, it's going to be it'll be increased resistance. Things to remember about this, there's this kind of idea like, oh, you're like eating that big fatty hamburger, like that fat's just kind of clogging up your arteries, and there is a correlation between, eating lots of saturated fats and having a higher incidence of these plaques, but it's not a direct relationship at all. The plaques are actually inside the wall of the vessel, right? We still have your tunica intima. The endothelium is still lining the walls of this thing. It's not like the fat is clogging inside the vessel and that's why it's clogged. This plaque is forming within the wall of the vessel. They're still understanding the full mechanism of how this seems to be an inflammatory process, turning on some inflammatory process, which results in this formation of this. And it obviously can be diet related, but again, it's not like the fats are just sticking inside your vessel. The fats are starting a much more roundabout process resulting in some inflammatory process, which results in the creation of these plaques within the walls, which then start impinging on the diameter of the vessel. So I'll leave it there. What else can we talk about? I should probably talk about shock just briefly. Right now, so usually we think of shock like, oh my gosh, but this is actually a clinical term for when you are not getting adequate blood flow to your tissues anymore. So you're not getting the, and obviously this is going to be really bad if your blood is not flowing out and perfusing your tissues. What are ways you can end up in shock where you have inadequate blood flow?
Hemorrhages.
What's that?
Hemorrhage. If you have a hemorrhage.
So yeah, so one kind
Bleeding.
of hypovolemic shock. Hypo meaning low volemic volume. So hypovolemic shock is if all of a sudden your blood volume is really low, which obviously means you don't have enough blood to carry things around. Plus, it's also going to totally mess with your blood pressure. Another way you can get this, if you think about 9 a.m. like in anaphylaxis, things that cause a major vasodilation. If you have really major vasodilation everywhere, and therefore you don't have the resistance, then you end up getting vascular shock. So this is like what's happening in anaphylactic shock. All of your blood vessels are dilating, the resistance is going down, your blood pressure is plummeting, and there's no longer pressure to keep the blood moving. So you can have the vessels cause it, you can have low blood volume. There's cardiogenic shock if the heart isn't working properly to generate the initial pressure to pump. So there's different ways you can go into shock. All of them need to be remedied pretty quickly or you're probably going to be dead. So anyway, just figure I should mention the idea of shock. What else? So now we are going to go to the final blood vessels. In some ways, the star of the show, because they're where the real kind of job of the cardiovascular system is happening. And that is in the capillaries. So capillaries, site of exchange between tissues and the blood. This might be delivering stuff like delivering oxygen and nutrients. It might be absorbing stuff like absorbing the nutrients from your digestive tract or picking up the hormones from a endocrine gland. But the basic idea is, this is where materials enter and leave the blood. There are 3 main types, and I'll talk about 2 super briefly and 1 in a lot of detail. So the continuous capillaries are the most common, and those are the ones I'm going to talk about in detail. But I should mention these other two just for completeness here. Fenestrated, it means having windows. So a fenestrated capillary has little pores. And these are extra super leaky. Right, a normal capillary's got to be kind of leaky or else things can't enter and leave. So a normal capillary actually is leaky, but a fenestrated capillary is extra leaky. You find them in places where, like in your, if you're trying to pick up hormones or something, or if there's a major amount of specialized stuff you're trying to bring bloodstream, you might find fenestrated capillaries. You also find on the opposite sides if you're trying to make, like when we talked about the choroid plexuses making cerebrospinal fluid, that happened because we had fenestrated capillary and fluid leaking out, that idea of filtration. So in filtration, you have H2O and small solutes leave. Again, we saw that was how we made CSF in the choroid plexuses. This is going to be a critical part of how the nephron works when we get to the kidneys. So you should understand this basic idea. The blood cells and the large proteins stay in the bloodstream. When I say extra leaky, you don't have the blood cells leak it out, or the really large proteins, but small solutes in water can pour out to make different kinds of secretions this way. So we will meet these again. Sinusoidal capillaries, these are where blood is being processed. In fact, let me just put it on a new page. These are in the liver, in the spleen, bone marrow. A sinus is kind of like an open space. So like when you get into the liver, you've got all these hepatocytes which are coming in and detoxifying and storing and doing things. You have white blood cells that are reaching in and recycling worn out red blood cells and cleaning things up. So sinusoidal capillary, they're kind of these big spaces. They often actually have breaks in the wall where like white blood cells can actually reach in and manipulate things. And they're usually very twisty, so the blood is going through slowly, so the surrounding cells have a chance to actually process things. So, blood is being processed. So sinusoidal capillaries. So, okay, so that gets us fenestrated, super leaky, used for extra absorption or else filtration to make, generate different fluids. Sinusoidal capillaries, specialized in these places where blood's being processed, where they're really twisty to slow the blood down. They are usually very super leaky, even so leaky they've got breaks in the walls so cells can reach in and process and clean up stuff in the blood. which will now finally bring us to the continuous capillaries. The most common, you're kind of generic. When you think of a capillary, is the one that you're normally thinking of as a continuous capillary. So let us talk about them. Right, so the basic idea here, you've got some venule coming in, not venule, some arteriole. The blood's coming in, feeds into some capillary bed. Some venule is draining it. You know, here's my cells. This is my tissue. So this is where the blood's coming in, the cells, you know, the oxygen can diffuse out the nutrients. That few things we need to add in here. The little precapillary sphincter. So the idea of a precapillary sphincter is it adjusts how much blood actually makes it into the capillary bed. Remember, I was saying that, like in your muscles, at any time when you're at rest right now, about 10% of the capillary beds are being used, and 90% are actually just lying in wait for when you actually are going to need more blood to your muscles if you're being active. So, you know, for most of your capillary beds in your muscles right now, the precapillary sphincter is closed off. You know, you still need some blood flow. There is actually these, they're called meta-arterioles. You know, so there is some minimal flow, so it's not like hitting a dead end. So there's still some, you know, blood that's going there, there's some flow, but in general, most of the capillary bed is not getting perfused. Like I mentioned, like in your muscles, you can increase the amount of capillary bed perfusion by like 10 times just by opening up more capillary beds. I talked about in your skin, there's a lot of that going on just for like taking, you know, heat and stuff. Capillary sphincter, even if you are opening up the capillary bed, it still doesn't just stay open with just, you know, stay open and let the blood come in. And instead there is a kind of pulsing. So it opens and closes, opens, closes, opens, closes, opens, closes, opens, closes. So you can see the blood cells kind of going in and kind of bumping through. It's probably just to keep things from clogging up, right? If you just keep kind of giving it little, you know, pushes, it makes sure that everything keeps rolling along. I should mention these capillary beds or the capillaries. Capillary is around 10 microns in diameter. That's about the same as the diameter of a red blood cell. So the red blood cells are coming in here. They're moving through single file. Right, which makes sense. If you think about, if you want to maximize like having oxygen, these are the ones carrying oxygen. We want them as close as possible to the cells of the tissues, because we saw diffusion works fine with really short distances, but not good on longer distances. So we want to get the cell carrying the oxygen really close to the cell that we're trying to deliver it to. Other things we can mention about these capillaries, they're pretty much pure endothelium. If you look at a capillary, it's basically, whoop, that's not where I want to go. So it's just these little frisbee cells making a super tiny tube. That's about 10 microns across, with red blood cells moving through, one at a time. We're not going to get into the detail of how things move across. Some of the stuff moves across actively, like transepithelial transport to get moved in or out. Some of it moves through tiny little clefts in between the cells. Just assume that things are exchanged. Oxygen and nutrients get delivered. Wastes get picked up for removal, things like that. The other thing that we need to talk about now, now I should not have made this meta-arterial green, because green is reserved for something else, which is going to make sense in a few moments. So I'm going to change the color of my meta-arterial. God, hold on. So it's time to think about hydrostatic pressure and osmosis. And like I said, some of these things that we talked about from the beginning of the semester are going to be with you until the very last day of this class. So let's talk about what happens as blood enters into the capillary bed. So I had mentioned a little earlier, that, as the blood starts at the high pressure coming out of the heart, as it gets into smaller and smaller and smaller and smaller vessels, it loses pressure because there's friction with the walls of things. By the time you are entering into the capillary bed, the blood pressure is about 40 millimeters of mercury. So that's like the mean arterial pressure as you enter into a capillary bed. At the other side, by the time you're leaving, going through these tiny, tiny little tubes, you're down to about 20 millimeters of mercury. And this is going to be important in a few moments. So now you have to remember these capillaries. You know, they're designed for things to enter and leave the blood, right? That's the raison d'être, is a site for materials to enter and leave the blood, which means even though they are not fenestrated or sinusoidal, they're still pretty leaky, right? That's otherwise things couldn't be coming in and out. So as the blood enters into the capillary bed here, you know, under a fair amount of pressure still, into these tiny little vessels with these super thin walls, water is going to start leaving. So as we enter into the capillary bed, we start losing water out of the plasma into the tissue space, that interstitial space. Right? I think I've mentioned that word before. The space in between the cells is just called the interstitial space.
So this is just osmosis?
No, it's not osmosis. This is hydrostatic pressure. There's quite a bit of pressure inside the vessels in the capillaries. And the capillaries are kind of thin-walled and porous, and water is leaving the closed vascular system just into the spaces between the cells, that interstitial space. However, as this is happening, so we're losing water. What is happening in terms of osmosis as we start losing more and more water, as we get further down to the end part of the capillary bed?
Osmotic pressure inside of the capillaries gets higher.
Exactly. So we're going to start having the water come back in at the far end because of two things. One, so we have, so H2O returns due to osmosis, and the lower hydrostatic pressure. So now there's not as much pressure as we're getting to the far side, pushing stuff out, but we have an osmotic pressure, which is driving the movement of the water back into the capillary. Right, so in the beginning, we are losing water due to higher pressure, but then we are then obviously creating kind of an osmotic gradient as we lose more water. So when we get to the far end, where we now have a higher osmolarity inside than there used to be, and now we don't have as much pressure pushing out, the water gets pulled back in due to the osmotic gradient. So does that make sense? And this is totally happening where as the blood enters into the capillary bed, fluid leaks out. And as it gets to the far end, things get sucked back in. However, over the course of a day, it's not perfect. So there is a net loss. Maybe I'll do it in black so it's really clear. So over the course of a day, if you were keeping kind of the accounting spreadsheet of how much water leaves coming into the capillary bed, how much water returns at the far end of the capillary bed, it doesn't match. You lose about 3 liters of fluid out of the vascular system every day in this process. Obviously, if we did not do something about this, your blood would turn to mud, right? If you just lose the water without getting it back in, your blood's going to stop working properly. So we need some way to reclaim that water and get it back into the bloodstream. What system do we have that can take that blood, that leaked fluid, and pull it back into the bloodstream?
Renal. No?
What's that?
Renal system?
It's not going to be the renal system.
The lymphatic system?
The lymphatic system. So the lymphatic system is going to come to the rescue here. So let me kind of introduce the lymphatic system, then we'll take our break here. So again, this is just a capillary bed going through my tissue. And like I said, 3 liters leaked per day. So, and I should mention this fluid here, we're going to end up calling it lymph once it's all pulled together. So one of the things I always kind of tell people is you're all used to the fact that oxygenated blood is usually represented as red. Vessels carrying deoxygenated blood are usually represented as blue, even if deoxygenated blood isn't actually quite blue. But lymphatic vessels usually are represented as green. So these are my little lymph capillaries. And they are just all over the place, permeating through your tissue, just like your blood vessels are. They're similar to capillaries, except obviously one big thing about them is, they're a blind alley. They don't start from any place. They just, it's regular capillaries. There's an entrance and there's an exit. Here, they just have these little things. And this is where this fluid and any other molecules or even cells, anything floating around in there can get taken up. And we'll talk about where it goes in a few moments. But these basically have the ability to absorb this fluid and kind of get it out of that interstitial space. A lymph capillary has these little endothelial cells that can actually kind of open up and flap. So as you're kind of moving around, these things are embedded in the connective tissue. These little endothelial flaps kind of open and close, and there's little suctions. So as you move around, it kind of drives the fluid and molecules or whatever floating around. Lymphatic capillaries, that little endothelial flap allows pretty big things to get absorbed into here. So I should be explicit about this. This is important for a couple of reasons. One is we are going to talk about the lymphatics being a critical part of absorbing fats from your diet. Fats come in, get absorbed from your digestive tract in a fundamentally different way than the other nutrients. And it's going to rely on lymphatic capillaries, the little lacteals, to take in these big macromolecules, these big lipoproteins and stuff. This is also important when we talk about, like, people who have cancer. Why do you end up taking out their lymph nodes and stuff? That'll be important in a few moments. So these lymphatic capillaries, they're all over the place. They're absorbing leaked fluid, any other kind of molecules and stuff that are floating around in here. Whatever that fluid is, once it's inside here, we just call it lymph. That's what lymph is. Lymph is just that fluid. And then ultimately, all of the lymph vessels ultimately come back. Usually, I mean, it's actually kind of more near the brachiocephalic vein is where they add in, but they basically dump all that fluid just back into the venous circulation. So all that leaked fluid that was leaving the bloodstream and the capillary beds ultimately is getting dumped back in. And again, this is not a great picture. You have a big, complicated system of lymphatic vessels and ducts and things like that. But the basic idea is all of this leaked fluid ultimately is just dumped back into the bloodstream. So now it's part of your blood again, which is kind of what you want. Fluid leaked out, we dumped it back in. Leaks out, you keep dumping it back in. Along the way, you go through these little things called lymph nodes. Which basically filter the lymph. So these, have what we have lymphoid tissue. Which are basically lymphocytes and macrophages, different kinds of white blood cells. So this lymphoid tissue inside the lymph nodes is basically filtering the lymph as it's percolating back to the heart. Lymphocytes and macrophages we are going to talk about in way more detail when we get to the immune system. So just for right now, just think of them as being kind of immune system, kind of protection, body defenses. Um, part of the reason when people have cancer, part of the reason why you worry about the lymph nodes, you've probably, like if somebody has breast cancer, they usually don't just get rid of the tumor, but they're going to get rid of most of the lymph nodes in the region as well. And that's because, cancer, what is cancer? Cancer is some cell that's dividing out of control. That's what the tumor is. The tumor is you have some cancer cell, some cell that has gone haywire and is dividing and dividing and dividing and dividing. If a cancer metastasizes, that's when you have something, part of it breaks off and then can start seeding a new tumor someplace else. That gets worse, right? And if you have cancer and the tumor staying local, at least you can just cut it out and you're done. But if it starts metastasizing, if pieces break off and start making new little tumors here and there, then you start fighting this losing battle. I talked about the lymph vessel having those flaps that can let large things as large as cells in. When a cancer metastasizes, it can get into the lymphatic system. And then that cancer cell is moving along and then it lodges in a lymph node and then starts just creating a new tumor there in the lymph node. So it's not uncommon when someone has a cancer that's metastasizing to have a lot of new tumors forming inside the surrounding local lymph nodes. So that's why they check the lymph nodes. That's why they often just remove the lymph nodes when somebody has cancer, to make sure that you didn't miss any little tumors forming there. Just briefly, this lymphoid tissue, I should mention, is lots of other places as well as lymph nodes. So let me just, for completeness, talk a little bit more about lymphoid tissue in the body. So it's in lymph nodes. This is kind of filtering the lymph. Another place you find it is in the spleen. The spleen is kind of like your lymph node, except instead of filtering lymph, it filters your blood. Right, if this is you, the spleen is kind of up here, right in the top left part of your abdominal cavity. And you've got an artery coming in, a vein coming out, the splenic artery, splenic vein. And the blood that is inside the spleen is getting cleaned up by those white blood cells. This is where you're recycling worn out red blood cells, stuff like that. So the spleen is basically a filter that is getting rid of worn out blood cells, as well as, kind of getting rid of anything else that shouldn't be there. Obviously, the spleen is one of these things that if you lose the spleen, you'll still be able to live a normal life, because your liver also does the same job. The liver also is a major place to filter blood and recycle worn out red blood cells and things like that. So the spleen isn't essential, but it's nice to have. It's also a place that I think stores platelets, and it also can act as a reservoir. Like if you are hemorrhaging, we talked about, hypovolemic shock, because you're losing all your blood. You can have the spleen contract and squish. Normally there's just blood percolating in here getting processed, but the spleen can squeeze and push more blood into circulation to try to keep your blood volume up. So the spleen can help you that way. And then this lymphoid tissue is also just, all along, walls of GI tract, trachea, etc. So if you think about I don't understand why this isn't. You've got your digestive tract going down into your stomach and all your intestines. You've got your trachea going down into your lungs. These are all these places where, some nasty little germ or whatever love to get its little claws into you. And you're sucking it right in, down your throat. You're swallowing it and going into these tubes in your body. You're breathing it in and it's going into this tube. So these are all vulnerable access points in your body. And so what you have are clusters of lymphatic tissue that are sitting along the walls there. You probably remember the Peyer's patches in the digestive tract. What do we call the big clusters right along, just on your throat? You have major collections of lymphatic tissue just along your throat, right underneath the skin.
Thymus gland.
Tonsils.
Tonsils. Tonsils are just lymphatic tissue. So this dude's coming in. All right, I'm ready to cause some trouble here. And the white blood cells here says, not so fast, you little pathogen, you. So that's another function of this lymphatic system, is kind of providing a barrier so where things are trying to cross the wall into your body, cross the mucosa into your body and get stuck by, or get blocked by the red blood cell, the white blood cells. What else do I want to say about lymphatic? I will just, again, a spoiler alert, which I mentioned a little earlier. I got rid of it. Because, so lymphatic vessels, lymph capillaries in the small intestine are for absorbing lipids. They're called lacteals. So I'm just kind of putting this here. So when we get to this later on, you'll know what's going on. Basically, like I said, the fats are tricky to absorb. Fat does not dissolve in water. So instead you have to make these large lipoproteins to move them around, but then those are too large to get absorbed into a normal capillary. So instead they're going to end up in a lymph capillary, which then is ultimately, we know, like, it's all going to be good though, because even if you're absorbing into a lymph capillary, ultimately, where does it end up?
Cisternochyli.
It's going to end up just dumped back into the systemic circulation. So, all right, so let's take a break. let's come back at 10:10. Okay, so I think what I'm going to do, yeah, I am going to finish up blood. It's the last thing we need to actually tie up the cardiovascular system. You know, there isn't that much to it because a lot of it we've already covered. And other parts we're going to cover. And also just kind of make sure we talk about the kind of the core things you need to know. All right, we are now going to finish up by talking about blood. We've already introduced it in the blood work lab. So some of this is going to just be a repeat. We talked about blood being a connective tissue, actually, with a fluid matrix, the plasma. And then the cells, which we call the formed elements. What do we call the ratio of formed elements to plasma?
Hematocrit.
Yeah, the hematocrit, which is typically about what?
Red blood cells.
But what's the value? What's the value of what percent plasma to percent formed elements?
It's like 45 to 50.
About 45 to 55, somewhere around there. So let me start with the plasma. Plasma is mainly water, like 92% water, maybe 1% dissolved, you know, kind of nutrients and gases, etc. Right? You've got sugar, glucose is in your bloodstream. You've got ions, you've got dissolved carbon dioxide, things like that. It doesn't take up that much volume, right? If I take a spoonful of sugar into a glass of water, the size, the volume doesn't change much. So you've got 92% water, 1%. The other 7% are plasma proteins. Excuse me. These are big proteins. These are the ones that tend to stay in the bloodstream even when there's filtration. Remember I was talking about like fluid leaking out of the vessels. The plasma proteins tend to stay back. Some of them you've met, right? You've met fibrinogen, like the one involved with clotting. There's actually lots of plasma proteins involved with clotting. You've met angiotensinogen, you know, involved with kind of the renin-angiotensin mechanism. There's going to be plasma proteins involved in your just maintaining basic osmolarity. So like one of the most common plasma proteins is albumin. Right, we know that we have to make sure that the plasma is around what, you know, 0.3 osmolar, or else we're going to have water if it was just pure water, the water is going to go out into the tissues and go into the cells. So we need to make sure that the plasma has the correct osmolarity. And this albumin protein is one of the most important players in maintaining that osmotic balance. There's alpha and beta globulins. Albumin, as well as these globulins, these act as carriers. Right, so there are things you want to move around in your bloodstream that are hydrophobic. There are fat-soluble vitamins. There are steroid hormones. So if you have something that you want to move around, a molecule that you want to move in the bloodstream, but that is hydrophobic, that doesn't dissolve in water, you're in trouble unless you got something to help you out. So these carrier proteins, these bind to these lipophilic molecules, and then as a unit can dissolve in the plasma and move around. We're gonna, a little later, we're gonna talk more about the big lipoproteins that are kind of the major ways to move, fats through the bloodstream, the high density and low density lipoproteins. But there's also a lot of small lipophilic molecules that get to move by being bound to these carrier proteins that allow hydrophobic molecules to actually dissolve in the plasma.
The uh, the the lipoproteins, they are cholesterol, right?
What's that?
Are lipoproteins cholesterol? Another
No.
name for lipoproteins?
Lipoproteins are combinations of lipids and proteins. We'll talk about them in more detail later. Plasma proteins we're going to see are going to be critically involved in immune response. So there's lots of them. Again, involved in blood pressure control, involved in immune response, involved in blood clotting. There's also one other kind. They're called gamma globulins, also called immunoglobulins, also called antibodies. And you've met these already, right? The antibodies, those were in the bloodstream. Those were part when we talked about the antibodies to the A markers and the B markers. But more generally, antibodies are parts of your immune response that we're going to see in more detail. So all of these, except for the antibodies, these are all made in the liver. So the immunoglobulins or the antibodies, they are made by lymphocytes, but all of the other ones are made by the liver. When we talk about the liver does so many different things, but one of the things the liver does is makes the plasma proteins. And this is actually, it's worth mentioning. In fact, let me, I think this is actually worth doing. We're going to talk about when when things go wrong, when fluid is not in the right place. So you can have really kind of tragic malnutrition and still be eating more than enough to kind of fill you up if you're not having the right balance. Like if all you're eating is bread, we're going to talk later when we do the digestive system, but if you're not getting all of the essential amino acids that you need to make all of your proteins, then your body does not have the ability to make all the plasma proteins that it needs, which case you're gonna not have the osmotic balance there. So I'll share. So kwashiorkor is a type of malnutrition where you're not getting all your essential amino acids, usually because you're just eating wheat, for instance, and not getting, and wheat does not have, it's an incomplete protein. And, you know, that's people sometimes like, how come people starving look like they're all bloated? And it's because of the fluid leaving out of not staying in the bloodstream and just kind of going out into the tissues and filling the abdominal cavity and stuff like that. So this is an example of what happens if you are not preserving that osmotic balance, for instance, in this type of malnutrition. Other thing I should mention, what happens if your lymphatic vessels aren't working properly? If the lymph vessels aren't draining that fluid that's leaking out of the capillaries into the interstitial space,
If fluid buildup.
Edema.
you get edema. Right, so here, this is an example of, this is this elephantiasis. This is this disease where you have this worm, this little parasitic worm that gets in there and starts blocking up the lymph vessels, scarring them up so the tissue is not draining anymore. So that fluid, instead of leaking and coming back and leaking and coming back, leaks and just stays in the interstitial space. And this is what's called edema, is when there is this buildup of fluid in the interstitial space. Again, you can see it's pretty dramatic. You know, I like to say, in fact, okay, so let me find. Yeah, so you can see lots of pictures of this. It's called, this is the official name of the disease. It's this little worm that gets in there. And you can see it can just get super intense. Like if the fluid is not draining out of the interstitial space, you can just get these horrible disfigurement like this. I can, maybe I shouldn't show. There's some really kind of, there's pictures of people literally moving their scrotal sacs around in a wheelbarrow because their scrotums have become so, I mean, it's probably worth just, you know, you can get horrible stuff like this, or in the legs. So, you know, when you think about the lymphatic system, you know, it's not just kind of like, you know, in fact, one of the old books by Marib was like, you know, the lymphatic system's not a superstar, but, you know, I'm like, when you see pictures like this, it's like, it's a superstar. It's doing something. You're very happy there. Part of the way you know you have edema versus just being kind of obese is if you have edema, and you push down. I can I show this here? If somebody's just obese, they just have a lot of fat underneath their skin, you push down, it's just going to spring back up. Fat is just kind of rubbery. If they have edema and you push down on them, then it's kind of like smart foam. You know, it's kind of like a Tempur-Pedic mattress. It stays down and slowly kind of comes back up. And again, if you've never experienced it, it's weird. I had somebody let me do it on them, and you know, sure enough, you know, you just kind of squeeze on them, and then you can just see your fingerprints in them. They just stay down in there. So, you know, again, so that's one of the hallmarks of this edema is if it's this fluid buildup in the interstitial space, if you squeeze down, the fluid kind of moves out. And then when you let go, it takes a little while to kind of squeeze back in. So anyway.
Besides the lymphatic problem causing the edema, the arterial congestion also causes edema, right?
Yeah, so exactly, like peripheral congestion, congestive heart failure will result in edema like that. You know, people will probably be on antidiuretics as well to try to get more water out of their body.
I mean, just diuretics, not antidiuretics, right?
Yeah. In fact, maybe this would be another one of these things, the edits, when I'll do. And so people will be on diuretics to kind of deal with that. All right, I'm going to go and we're going to finish up the blood with the formed elements. There's going to be WBCs, the white blood cells, also known as the leukocytes. The RBCs, red blood cells, also known as the erythrocytes, and the platelets. Sometimes platelets are called thrombocytes, but I don't even want to say it because they're not, that means cyte means cell, and platelets aren't really kind of viable cells. All right. We'll start with the white blood cells. White blood cells are involved in immune response. We are going to talk about them in much more detail when we get to the immune system, the neutrophils, the monocytes, the eosinophils, the basophils, the lymphocytes. Don't worry about the details of them right now. Main thing is immune response, and also they are not confined to the closed circulatory system. In fact, you know, most of your white blood cells are not in your bloodstream. They're like hanging out in lymph nodes or just patrolling tissue spaces or doing all sorts of stuff. And even the ones that are in your bloodstream, they'll get a chemical message if there's some infection happening, and they'll grab onto the wall of the blood vessel and they'll just move out of the vessel into the tissue spaces to go fight the battle. So these white blood cells, you know, while you can think of the cardiovascular system as kind of a staging area, and you know, they are also just in your tissue spaces and the lymphatic system. They're all over. So, and again, we will talk about them in more detail later. RBCs, these are most common, like 99% of the cells. Red blood cell, let's talk a little bit about them. When you look at them, again, you've already looked at them in pictures in class. They tend to look kind of this little, we call a biconcave disc. You know, from the side, they look kind of like this. From the top, they look like this. And that's because they eject their nucleus. So during development, you know, they in all of these things are made in the bone marrow. The bone marrow is where you have hematopoiesis. During hematopoiesis, you have these cells with lots of, they have a nucleus, they have lots of rough endoplasmic reticulum, they're making tons of hemoglobin. But by the time they're mature and on patrol, these are, there's no nucleus. They contain about 250 million molecules of hemoglobin. And no mitochondria. So let's talk a little bit about this. No nucleus. What does that mean? If it doesn't have a nucleus, what is it not going to be able to do?
DNA, cell replication.
It can't divide and it can't even repair itself. It can't make proteins anymore. So once you have this thing off in the circulation, it doesn't have very long to live because it's going to wear out and it cannot be repaired. So, you know, only live, up to like 4 months at most. So they're constantly being recycled. They're recycled in the liver and the spleen. We should spend a little bit of time now talking about the details of the hemoglobin. And again, no mitochondria because their job is to carry oxygen. So we don't want them using up oxygen. Plus, they don't have much metabolic activity that is going to require lots of ATP. They don't do much other than, they do some stuff, but they don't do a lot other than carry oxygen around. So they can get the ATP they need just by, glycolysis. So no mitochondria. So let's talk about hemoglobin. It is a big multi-part thing. It's got these protein, they're called globin subunits. So this is actually, it's a protein. Having subunits means that it's got that quaternary, that fourth order structure. So globin subunits, and then there is these iron containing heme groups. Each heme group binds an oxygen. The globin subunits, on the other hand, have binding sites. These can bind carbon dioxide. And whoops. So here's my entire hemoglobin here. This is one hemoglobin. one hemoglobin molecule. It has two jobs. Its more famous job is it binds oxygen, but it also is important. It's going to end up carrying about 30% of your CO2 in your bloodstream bound onto the globin subunits. So that is actually an important part of what it does as well. So each hemoglobin has 4 heme groups. Each heme group combine an oxygen. So how much oxygen can you put in one red blood cell?
250 million.
Those 250 million molecules of hemoglobin
Be a billion.
be a billion. So I was like 1 billion molecules of oxygen in one red blood cell, which is kind of crazy. The carbon dioxide is also bound there. And we're going to see when we get to the respiratory system later and talk more specifically about gas transport, the affinity of hemoglobin for oxygen is altered by how much carbon dioxide is bound on the hemoglobin. So we're going to see this is actually going to be important. It's called like the Bohr effect. This is not going to be something for right this moment, but the next class we're going to talk about the affinity. Or maybe let me write it out though, just so you have it's percolating in your head for a little later. Hemoglobin's affinity for oxygen. Let's put it affected by whether or not carbon dioxide bound as well. So we're going to talk about this in more detail. But basically, hemoglobin can hold on tighter or less tight, let oxygen go more easily or not, depending on how much CO2 is around. And we'll talk about why that makes sense. I should also talk about carbon monoxide. Right, carbon monoxide, that's not CO2, that's CO. This is a deadly poison. This binds with 200 times the affinity of oxygen. So why is it a bad idea to fall asleep in your car with your garage closed with your engine idling?
You will die from carbon monoxide poisoning because it will bind to hemoglobin instead of the oxygen.
Exactly. So this carbon monoxide, it grabs onto the heme group. And even if there is oxygen around, doesn't matter because this is holding on 200 times tighter. So that means no oxygen is getting transported to your tissues and you'll die. Ironically, hemoglobin normally, so, oxygenated hemoglobin is bright red. It's kind of more dull, bricky red or looks blue through your skin. So normally when somebody is not getting enough oxygen, you can tell because they start looking blue. We say cyanotic. Cyanotic meaning bluish. So if somebody suffocates, usually you see them and they're going to be blue. However, if somebody dies of carbon monoxide poisoning, because actually the hemoglobin is totally bound onto this carbon monoxide, they're dead, but they're like kind of bright cherry red. They look even redder than normal, like flush, because the hemoglobin actually is bound, but it's bound to something that's not allowing the oxygen to flow or to bind. But this is actually an important diagnostic, realizing that hemoglobin, when it's bound to oxygen, is bright red. And so if somebody has diminished oxygen in their blood is not going to, they're going to look more kind of pale or even bluish. And there's places, even, obviously, depending on the color of your skin, it might be harder to see whether or not somebody is turning blue or not. But there are kind of places you can look where there's not as much pigment in the skin. So even if somebody has naturally more pigmented or darker skin, you can still kind of check for this cyanosis, this kind of bluing thing going on. Okay. So we are at the this looks like 12:30, so it's the end of our week. I still do want to talk a little bit more about blood clotting, but that will have to be on another day.